Hi everyone, today we are diving into a complex case of carbohydrate metabolism. Our patient is a 50 years old male experiencing chronic fatigue and weight loss. Lab results show fluctuating blood glucose and elevated lactate levels. Let's see what's happening at the cellular level. First, let's talk about the Krebs cycle. The Krebs cycle plays a central role in cellular energy metabolism. It acts as the final common pathway for the oxidation of carbohydrates, lipids, and proteins. In the cycle, acetyl-CoA is oxidized to produce carbon dioxide, along with reduced coenzymes such as NADH and FADH2. These coenzymes are very important because they carry electrons into the electron transport chain, where large amounts of ATP are produced. Therefore, the Krebs cycle is essential for efficient energy production. In addition, the Krebs cycle is um, amphobiolic, meaning it also provides intermediate for biosynthetic processes, such as the synthesis of amino acids, nucleotides, heme, and even for gluconeogenesis. The connection between glycolysis and the Krebs cycle occurs through pyruvate. Pyruvate must first undergo oxidative decarboxylation by the pyruvate dehydrogenase complex to form acetyl-CoA before entering the cycle. In this patient, the increased lactate level indicate that pyruvate is not optimally entering the Krebs cycle and instead is converted into lactate. This result is reduced ATP production and contributed to chronic fatigue. The disruption of the Krebs cycle leads to a significant decrease in ATP production. Under normal aerobic conditions, one molecule of glucose can produce approximately 30 to 32 ATP. However, when the Krebs cycle is impaired, the oxidation process is incomplete, and the cells shift to anaerobic metabolism. In this condition, pyruvate is converted into lactate to regenerate NAD+ so that glycolysis can continue. This pathway produces only 2 ATP per glucose molecule, which is far less efficient compared to aerobic metabolism. As a result, the body experiences an energy deficit, leading to chronic fatigue. The increased lactate level observed in this patient also indicate that the cells are operating under conditions similar to anaerobic metabolism. Lactate accumulation not only reflects inefficient energy production, but can also disrupt cellular function. Additionally, the decrease in enzyme activity further inhibits the Krebs cycle, since each step in the cycle is catalyzed by a specific enzyme. Any reduction in enzyme activity will slow down or even stop the entire cycle. Gluconeogenesis is a crucial process that ensures a continuous supply of glucose, especially for the brain and red blood cells during fasting conditions. This process is regulated by hormonal signal. Glucagon stimulates gluconeogenesis when blood glucose levels are low. By increasing the activity of key enzymes, in contrast, insulin inhibits this process after meals to prevent excessive glucose production. Cortisol also plays an important role by increasing gluconeogenesis during stress. It mobilizes amino acids from muscle and fatty acids from adipose tissue are to be used as substrate. In addition to hormonal regulation, gluconeogenesis is also controlled by the energy status of a cell. High ATP level promote gluconeogenesis, which high AMP level inhibit it. This regulation ensures that gluconeogenesis only occurs when sufficient energy is available, since it is an energy-consuming process. The Cori cycle is an important metabolic pathway that connects muscle and liver metabolism, allowing the body to recycle lactate into glucose. During intense physical activity, muscle cells rely on anaerobic glycolysis to produce ATP quickly. In this process, pyruvate is converted into lactate. This lactate is then released into the bloodstream and transported to the liver. In the liver, lactate is converted back into pyruvate and then enters gluconeogenesis to form glucose. This glucose is released back into the bloodstream and can be reused by muscle cells as an energy source. In this patient, the evaluated lactate level indicate that the rate of lactate produced exceeds the rate of its conversion in the liver. This is likely due to impaired liver function, which reduces the efficiency of gluconeogenesis. As a result, lactate accumulates in the blood, contributing to fatigue and potential metabolic imbalance. The pentose phosphate pathway is an alternative pathway of glucose metabolism that primarily serves anabolic function rather than energy production. Its main products are NADPH and ribose 5-phosphate. NADPH is essential as a reducing agent in biosynthesis process, such as fatty acid and steroid synthesis, and also plays a critical role in protecting cells against oxidative stress. Ribose 5-phosphate, on the other hand, is required for the synthesis of nucleotides and nucleic acid, making this pathway important for cell growth and repair. Therefore, even though PPP does not produce ATP directly, it is essential for maintaining cellular function and biosynthesis. The pentose phosphate pathway is particularly important in tissue such as the liver and adipose tissue, where lipid synthesis is highly active. In this tissue, NADPH produced by PPP is required for fatty acid and cholesterol synthesis. NADPH provides that reducing power needed for this biosynthesis reaction. Without sufficient NADPH, that synthesis of lipids will be impaired, affecting energy storage and membrane formation. In addition, NADPH is also important for maintaining redox balance by regulating reduced glutathione, which protects cells from oxidative damage. In this patient, impairment of this pathway might contribute to weight loss and reduced lipid synthesis. Gluconeogenesis is the process of converting glucose into glycogen for storage, mainly in the liver and skeletal muscle. The process begins with the phosphorylation of glucose to glucose 6-phosphate, which is then converted into glucose 1-phosphate. This molecule is activated into UDP glucose, which serves as the direct substrate for glycogen synthesis. Glycogen synthesis that elongates the glycogen chain, while the branching enzyme forms alpha-1 linkage to create a brace structure. This process occurs primarily in the feed state when blood glucose levels are high and insulin is released. Case one is patient weight loss suggests a failure in energy storage. If gluconeogenesis is insufficient, the body lacks backup fuel during fasting. Gluconeogenesis is the process of breaking down glycogen into glucose to maintain blood glucose level during fasting or energy demand. This process is initiated by glycogen phosphorylase, which cleaves glycogen into glucose 1-phosphate. This is then converted into glucose 6-phosphate. In the liver, glucose 6-phosphate is converted into free glucose and released into the bloodstream to maintain blood glucose level. In contrast, muscle cells lack the enzyme glucose 6-phosphatase, so glucose 6-phosphate is used locally for ATP production. Glycogen metabolism is regulated by the hormones insulin and glycogen through opposing mechanisms. Insulin promotes glycogen synthesis by activating glycogen synthesis and inhibiting glycogen phosphorylase. This occurs during the feed state when blood glucose levels are high. Glucagon, on the other hand, stimulates glycogen breakdown by activating glycogen phosphorylase and inhibiting glycogen synthesis. This occurs during fasting. This opposing action ensures that blood glucose level remains stable under different physiological conditions. Case analysis is the patient unstable sugar despite fasting, indicate a broken switch. Liver dysfunction makes it less responsive to glycogen signals. Carbohydrate metabolism is a highly integrated system where multiple pathways are interconnected to maintain energy balance. Glucose 6-phosphate acts as a central metabolic hub. It can enter glycolysis for energy production, glycolysis for storage, the pentose phosphate pathway for biosynthesis, or gluconeogenesis to maintain blood glucose level. The Krebs cycle is linked to gluconeogenesis through intermediates such as oxaloacetate, while the Cori cycle connects muscle and liver metabolism. In this patient, disruption in multiple pathways, including impaired Krebs cycle, ineffective Cori cycle, and reduced gluconeogenesis, lead to decreased ATP production, lactate accumulation, and unstable blood glucose level. This demonstrates how failure in one pathway can affect the entire metabolic network. In conclusion, carbohydrate metabolism is a highly integrated system involving multiple interconnected pathways, including the Krebs cycle, glucogenesis, glycogen metabolism, and the pentose phosphate pathway. Each pathway plays a specific role, but they work together to maintain energy balance, regulate blood glucose level, and support cellular function. In this case, disruption in a separate pathway leads to decreased ATP production, lactate accumulation, and unstable blood glucose level, which explain the patient's chronic fatigue. This shows that even a small disturbance in one metabolic pathway can affect the entire system. Thank you for attention.